Welcome to Solutions OP Clinical Trials Podcast. My name is Olga and I'm the Managing Director of Solutions OP. We are a small team of experienced professionals working on clinical trials in the United Kingdom. I'll use our podcast to discuss hot topics in clinical trials and provide analysis of the latest trends and challenges in pharma and medical device industries, not just in the UK but globally. Thank you for listening. Welcome to today's episode of the podcast. Today I'm joined by my colleague Lydia Ainsworth, who is our Senior Clinical Data Quality Manager, and we'll be talking about animal testing and more specifically the purpose of animal testing, the ethical issues, and also the future of animal testing. It's a very popular topic at the moment. Among many regulators, everyone's trying to reduce the amount of animals used in clinical research. So I think will be a very interesting topic for everyone. So animal testing existed throughout the centuries. It's not a new concept. And initially, the animals were used to study anatomy and physiology, but also to help the development of medicine. And animals were used for to study human diseases. However, dissection of animals is not always the best approach of studying diseases. So why? That's because when you have an animal which is dead, then that changes the processes and the chemistry of the tissues. And it's not always the best way to study the disease or to actually get appropriate information about the physiological process. Apart from animals, during the Renaissance, they started using human cadavers for studying anatomy and physiology, but still animals are used even until today for studying and modeling different diseases and learning different developments of new drugs. So Lydia will talk a little bit more later on about the ethical issues of using animals in testing and the alternatives to animal testing, and I will cover some other aspects. Another thing that I wanted to mention here is it's not only the ethical problem with using animals, but it's also the cost and the extensive testing period that you need to obtain all the necessary clinical data for the study. And as I mentioned, animals are not always the best models and the most reliable models when you want to study human diseases. So I'll start with the first part, which is why do we need animal testing and in clinical research, and what animals are the ones that are used more frequently. I think it's very important to kind of give a respect to the contribution that animals have done to developing of human medicine and how we study the human diseases. The most common animals used in clinical research are mice and rats, and they're approximately 95% of the animals used in clinical research. They're preferred because they're small and easy to house, and they have a short reproductive cycle, and their lifespan is 1 to 3 years, so they're kind of easier to maintain. But also they are relatively easy for any genetic manipulation, so that allows to create mice that have different genetic diseases and allow to study different diseases which are observed in humans as well. And also their organs are smaller in shape and structure, and they're kind of similar to human organs. So mice were used as models for many diseases like hypertension, seizures, cancer, obesity, heart diseases, respiratory diseases, deafness, and many other conditions. And also mice are used to study behavior changes, nutrition, aging, sensory, and also for some genetic studies. The other type of animals which are quite often used are rabbits. So it's very interesting that the world first vaccine for rabies is actually developed in rabbits as test animals. And rabies, if you don't know, it causes acute encephalitis. So it's a very serious disease, and it's a very important vaccine that was developed. So rabbits, they have some special bred rabbits used in clinical research. There's with specific diseases, and they're used normally to study myocardial infarction and arteriosclerosis. But also rabbits are used to study human pregnancy and also developing monoclonal antibodies for immunology research. The other animals which are probably the second most common used in clinical research are dogs, interestingly. After mice, normally they use dog models, and it's quite common, I think, we see in oncology studies. They've used dogs as a second type of animals for toxicology testing. The reason for that is because the data obtained from dogs could be extrapolated to humans, and that's why they're preferred. But also, dogs are used to study liver toxicity. They're a very sensitive model. And the last group of animals which are used in animal testing are monkeys. So they are used to develop new drugs and medical devices specifically for treatment and prevention of infectious diseases. However, sometimes they are also used to study eye diseases and transplantation research. So monkeys are used only as like a backup plan if there are no other reliable animal models that could be used. Normally, they're used to study drugs that cause birth defects or drugs that affect the central nervous system, or for testing monoclonal antibodies, or also to test drugs which cause vomiting or any conditions. So because the testing on monkeys could predict hazard on humans, they are used also for pediatric research, so for anyone developing drugs in pediatric patients. But as I said, apart from the ethical reasons for not using animals, there are other reasons, and cost is one of them. So my research showed me that the cost of mice per animal is between $20 and $300 and that depends. So genetically modified mice, they tend to be more expensive. For rabbits, that cost is between $125 to up to $270, again depending on if they are special bred rabbits for a particular disease. For dogs, the cost is between $300 up to $1,000, and again it depends if you have any special breed. For monkeys, that cost go even higher. It's $6,000 per monkey, and for some special monkeys which are bred for research, that cost can go above $50,000 per animal. And that's only for the animal costs. Otherwise, for housing for smaller animals, it's between $1,500 per year for housing and caring. And for animals, that for monkeys, that cost can go up to $10,000. So it's apart from the ethical reasons, it's very expensive, and creating these special breeds also, it's an expensive process, and that's why we need to find another solution. Even though we acknowledge all the benefits of for medicine from using animal models, we need to find some alternative solutions and think about the ethical issues that we have from using animals in research. So Lydia will cover Now, the other part, which is what are the ethical issues with animal testing in clinical research?
Thank you, Olga. So as Olga said, I'm going to discuss some of the ethical issues with animal testing and clinical research. So the ethical issues of animal testing come down to how you view the moral status of animals, so their intrinsic value, and how we as humans understand their pain and life experience. Obviously, animals can't give consent in a way that is required by humans to participate in research. So because of this, there have to be a lot of ethical regulations in place to minimise the suffering caused to the animals. But going back to earlier research practices, the use of animal testing wasn't regulated because it was believed that animals didn't experience pain in the same way as humans do. But it's now well documented in animal behaviour research that animals do experience pain and distress, and the procedures used in animal testing might subject them to this. So this includes physical distress from the experimental procedures themselves, but also psychological distress from lab conditions, which might result in social deprivation and an inability to display natural behaviours as they would in the wild. So the use of animal testing in research creates a conflict of moral interest, because if on the one hand, if you were to argue that animals have the same intrinsic value as humans, their use in clinical research would not be justified at all. However, in clinical research, the ethical decisions about the use of animals are made from a utilitarian perspective, which means that the use of animals in research is justified if the benefits to human beings outweighs the harm caused to the animals. And weighing up the harm against the potential benefit can be difficult. So, as I said before, there's quite a lot of regulations on making these kind of decisions. So, for example, the European Medicines Agency recommends the 3Rs as guidelines for assessing ethical decisions for research involving animals. So this includes reduction, so reducing the number of animals involved in experimentation to the minimum required to have reliable data. Then there's refinement, so the procedures have to minimise the harmful effect on animals. So for example, that can include things like making sure staff are properly trained and using particular methods which reduce suffering. For example, things like anaesthesia. The third R is replacement, so where possible, researchers have to use alternative techniques that don't involve animals at all, and I'll talk more about that in a moment. And some researchers and within regulation are also talking about the fourth R, which is responsibility. So researchers have a responsibility to promote animal welfare and commit themselves to understanding and researching animal suffering to further improve the conduct of research that involves animal testing. So these guidelines emphasise that the use of animals in research requires careful consideration and calculating many different factors to make sure that harm is kept to a minimum while the potential benefit is maximised. But obviously this comes with some issues. So one issue is that at the stage of animal testing and drug development, which happens very early on, there's often no proven benefit to humans at that stage. So animal models can only provide us with so much information, and drugs or interventions might prove to be successful in animal experiments but unsuccessful in humans, meaning animals might suffer without a guaranteed benefit for humans. And on the other hand, some drugs might have been withheld from humans because they weren't effective in animals, but they were later proven to be safe and effective in humans. So one example of this is aspirin. So in early animal testing, it wasn't shown to be effective, but now it's obviously approved as a really safe and effective drug in humans. And this goes to show that animal testing isn't always reliable, and it's not always guaranteed to be ethical, despite following the procedures for maximising the benefit to humans while minimising harm to the animals involved. So because of this, all research involving animal testing has to undergo in-depth ethical reviews, and this is the case for regulatory authorities across the world. So the UK Research and Innovation, the European Medicines Agency and the FDA all have this review process for any research involving animal testing. And the researchers conducting these tests also have to undergo very strict regulations and measures.
Thank you, Lydia. Thank you. That's brilliant, and it's very good points about the ethical issues, particularly with the pain. The way that was accepted, there was no pain, and sometimes yes, we don't know at the early research if that actually is going to help humans. So these are very good points. And it's a very good transition to my next part, which is about in what cases it's acceptable to use animals in clinical research, because there are some cases where we need that and it's a proven benefit for humans. So, for example, there are diseases which are observed in animals which are also observed in humans. And then in that case, it could be acceptable to use the animals as for the developing of new treatments. And the best example for this is the simian immunodeficiency virus, SIV, which is naturally observed in animals. And it's a similar development to HIV virus. The SIV is observed in gorillas, chimpanzees, and mangabeys, and it was critical that the fact that scientists were able to work with these monkeys and study SIV and learn about the natural history of SIV. It helped a lot in development HIV therapies and treatments later on. For example, monkeys were used to study the transmission of the virus from the mother to the baby, by taking blood from them to see how the virus transmits. But as I also said, we wouldn't have the initial anti-HIV therapies without the monkey research back in the day. So they were really critical for this. Another part where animals have been really important is to achieve transplant tolerance. And this means to train the immune system not to recognize the new organ as foreign and attack it. So transplant tolerance could be different between the different species. So what works in mice may not work in pigs, which is, kind of something we can guess. So the scientists have developed this method, and it's particularly used back in the days for kidney transplants. To create this transplant tolerance, they started from mice and then they went through pigs and then monkeys and humans so that they can transplant the kidney to the humans. So the transplant, the tolerance is built when you transfer donor stem cells alongside the kidney. So this is to train the immune system to accept these cells as its own, and gradually by using these stem cells from different animals to help actually create a transplant which could be acceptable for the human. Another important part where for example, monkeys are used is in developing, in the studying and developing psychiatric conditions and neurological diseases like Parkinson and dementia. So they do a lot of observational studies to see how this affects monkeys and do extrapolations for humans. And another important part where, again, they've used monkeys is for vaccine studies, to study immune response to antibodies or to viruses, because there are a lot of viruses which affect monkeys, which are similar to the viruses that affect humans. So if they have blood samples that they can test and learn from them, that could help understand how the virus develop in humans as well. So it's there are cases, as I mentioned, where it's been critical to use animals, and it's probably at the moment there is no alternative. But again, that doesn't mean that there will be no alternative in the future. And here we come to the next section where Lydia will tell us about what alternatives are available, and maybe we won't need to use animals in the future. We'll learn more from her.
Thanks, Olga. Yeah, it definitely seems like there is more of a push recently to move away from animal testing to other methods. And obviously that's kind of come with a lot of advances in technology and research techniques. And there's a lot of also investment in these methods because, as you discussed earlier, Olga, animal testing can be really expensive. So wherever companies can reduce the use of animal testing, they seem to be eager to do that. So some of the alternatives include in chemical methods. So researchers can perform lab experiments on proteins and DNA outside of human cells. So this can be used to look at drug interactions at a molecular level and give some indication of how this might affect the human body without the need to test on live animals. You also have in vitro methods, so this includes things like tissue culture and 3D printing techniques, which allow researchers to grow human cells outside of the human body and even in some cases produce mini organs which can replicate human organs and the physiological processes. So again, these can be used to test how human cells might respond to a drug and might be able to be used instead of animal models to provide indications of how a drug behaves and can provide accurate information on drug toxicology without the need for animal testing. There's also a lot of advancement in what's known as in silico methods. So this includes things like AI and computer simulations. So some advanced AI modelling can simulate how compounds might interact with the human body. So that uses existing information of the investigational drug or compound, information about biological processes, and uses mathematical predictions to model how that might work. And this can be faster and often cheaper than animal testing. So like I said before, it can be appealing to drug development companies. But yeah, like Olga was saying, obviously sometimes animal testing is still needed. So these alternatives aren't without limitations, and it's unlikely that they will be able to replace all animal testing procedures. So animal testing might still be required in some areas of clinical research. But I'd say that overall it seems like within the field of clinical research, there seems to be a lot of focus on the reduction and replacement guidelines within the 3 Rs, like I talked about earlier. So moving away from the use of animal experimentation where possible to minimise unnecessary suffering while still promoting medical advancements and the improvement of human lives. And last year, the UK government produced a press release, I think it was in November, which outlines some of the strategies that it intends to implement to phase out animal testing in favour of some of these alternative methods. So it does seem like we are moving away from animal testing and taking advantage of these alternatives, which are becoming a lot more available.
Yeah, and that's very true. And it's not just the UK MHRA. FDA also very hot on the topic of moving away from animal testing, and they're really in favor of this. I think particularly in the UK, we've seen the in silico models are very strong. I actually attended a few calls where they were discussing how you can use in silico models to predict and extrapolate data and predict how the disease will develop and your endpoints and so on. So it's very interesting method. Maybe you're right. Maybe you can't use it completely to eliminate animal research. But again, we don't know how that will progress with the AI and if we'll be able to have, for example, any AI models that we can use that could actually provide us more accurate and better data in the future. So. I think it's still open to many new opportunities for everyone who wants to contribute to research and reduce the reliance on animals, which I think is a very good thing. If we can remove the need to use animals, that would be fantastic. But at the moment, we are not there yet. So thank you so much for listening. I hope it was an interesting one.